Okay, in this video, I'm going to go over chapter 8, and it's a long one. We're going to go over transcription, translation, and protein processing. So, pretty much the conversion from DNA to RNA, and then RNA to protein, and also how those proteins get processed, and if they need to be secreted out of the cell, we'll also talk about that. So there's a lot going on in this chapter. So we're going to talk about RNA polymerase and how transcription happens. Then we'll talk about translation, ribosomes, all of that. And then we'll talk about the modification of proteins, how they fold, and how the bacteria secrete them. All right, so let's get into it. 8.1 goes over the RNA polymerase. So this is the enzyme that obviously transcribes DNA to RNA. So it's also called the DNA-dependent RNA polymerase. DNA-dependent meaning that the template that it uses is DNA to make RNA. And this is what we call the core protein, and it's the main protein that performs transcription. So there are about 5 subunits that are found in bacteria. So we have the alpha 1, you can see it here, and then there's alpha 2, and then there's beta prime. Those are the main 4, and then we also have an omega subunit. So there are 5 main subunits for the RNA polymerase in bacteria. Now, the RNA polymerase also usually binds to these sigma factors. So these are additional proteins that help guide the RNA to the beginning of the gene. So by itself, this is not able to find the beginning of the gene. It is able to do transcription, but not actually locate where it should be going. And so the sigma factor is what helps do that. So these are all sigma factors here, and they would bind and kind of help find the location on the DNA where it should start. And so together, the RNA polymerase plus the sigma factor is called a holoenzyme. So a holoenzyme is the combination of the polymerase itself plus the sigma factor. Okay, so how does this whole complex figure out where to start or where to begin on the DNA? It uses promoters. So promoters are regions of the DNA upstream. So upstream means before the gene itself, and it's going to be where the holoenzyme binds. So basically, it's where the RNA polymerase binds to the DNA. And so this is just some examples of promoters that are found in E. coli. And so +1 means that this is the beginning of the transcription start site. So this is where transcription would begin. And so all of this doesn't get transcribed. It's just where the RNA polymerase will bind. And then it will kind of move over and then begin transcribing here. So this is just the binding region, essentially, of where RNA polymerase is going to go. Now, most of these promoters are found 35 and 10 nucleotides before the beginning of the transcription start site. So again, this is the beginning, and so it's -10 and -35 approximately. These are where the promoters are. But usually people just call this entire chunk the promoter region. Okay, cool. And so let's talk about these sigma factors again. So there is one very important sigma factor in E. coli called the RPOD, and that's also known as the sigma 70 or sigma factor 70. And this is the most common sigma factor in E. coli. It pretty much activates most genes. So most of the genes that are important for E. coli get activated through this, not activates, but this is the one that it binds to. So RPOD binds to most of the genes that E. coli uses. There's obviously other ones, so RPOS, and these guys help with turning on certain genes that are more specific. So RPOS, for example, turns on genes so that the bacteria can go into stationary phase. Or RPON turns on genes that have to do with nitrogen metabolism. So some of these sigma factors are specific to certain types of genes, but the RPOD is the one that's like for most of the regular genes that are always active in E. coli. Okay, and so how does it bind? So here is the holoenzyme, and you can see the sigma factor is like kind of here at the top, and the -35 and -10 region of the the DNA binds to this sigma factor, and so that's how they bind, and you can kind of zoom in too. And so that's why it's important that the promoter region is at -35 and -10, because it has to fit perfectly with the sigma factor. Okay, so now that we've kind of established who is responsible for transcription. Let's talk about what a gene is. So this is a diagram of a gene. And as you can see, there's that promoter region, -35, -10, and this +1 is this transcription start site. This is where the RNA polymerase will begin to create RNA, or it'll read it and make the RNA starting from here. And so some things to recognize is that there is an untranslated leader, and that is on the mRNA. And again, it's called untranslated because translation will begin here. Okay. Just like how there's this extra space here, even though transcription starts here, there's this extra space here, and then translation starts here. So just like how the promoter is the binding site for the RNA polymerase, there has to be a binding site for the ribosome that happens before it actually starts. And so this is where the ribosome binding site is. It's in the untranslated leader region. And so the way I generally think about it is like when you're starting a race, right? You have like the start line, but everyone is just slightly before the start line, right? If you're thinking of like a track and field race, everyone is literally one step before the line where you start, right? And so that's pretty much it. So this is the start line for transcription. This is the start line for translation. And there has to be a region where the enzyme for RNA polymerase binds here. And then for the ribosome, it needs to bind somewhere. So there has to be somewhere before the start line where it's hanging out, right? And so that's the way I think about it. So yeah. So, like, I guess I could just draw really quick. So, how like on track and field, there's like a line, and then all of the people are like standing here, right? And then they blow the whistle, and then everybody starts, right? So we're like right before the start line. Okay. Anyway, so that's the untranslated region. It contains the ribosome binding site. And then at the very end of the DNA, basically right past the last gene, you're going to find a transcription terminator region, and this is going to stop transcription. So this is the finish line, essentially. And notice that there's also a finish line for translation, because we need to know when the ribosome needs to know when to stop translation, and the RNA polymerase also needs to know when to stop transcription. So I always think about it like a race. There's a start line and there's an end, and they're different for the RNA polymerase, and it's different for the ribosome. Okay, cool. So let's now talk about transcription, how it begins. So this is initiation, the first step of transcription. So first thing that happens is the hollow enzyme. Remember, the hollow enzyme is the RNA polymerase plus the sigma factor. So there they are, as you can see, with the sigma factor. And they're going to scan, basically, and look for a promoter region. So it's going to look for that sequence that it recognizes at the -35, -10 region. And if it recognizes it, then it's going to bind. So that's what it does. The whole thing binds to the promoter, and it closes, meaning that it's now in the closed form. So it's now ready to transcribe. And so last step is that the ribonucleotides, so the nucleotides that make up RNA, are going to come, and it starts to make mRNA, right? So mRNA is being made. And the sigma factor comes off, because really the sigma factor's only job was to find the promoter. Once it finds it and transcription begins, now it's just the RNA polymerase by itself. The sigma factor can be reused for something else, to bind to a different promoter. So this guy gets recycled. Okay, cool. So now transcription is happening. So the mRNA is being made. And then at the very end, we have to stop transcription. So just like a race, there's the start and then there's the stop. So how do we stop? There's two ways. So there's row-dependent termination. So row-dependent means that it depends on this protein called Rho. So Rho is a protein binds to a specific region on the mRNA. So you know how this RNA polymerase is making mRNA, and the mRNA is kind of like floating around, right? Still attached to the whole complex, but there's a little strand of mRNA coming off. And so the Rho protein binds to this newly made mRNA molecule, a little bit over here, not so close, but it's here. And it basically zips along the mRNA and gets closer and closer to the RNA polymerase. So it just kind of drags itself along until it gets closer to the RNA polymerase. And then once it touches the RNA polymerase, this Rho protein has it basically has like a helicase function where it can unwind molecules of nucleotides. And so what it's going to do is unwind the RNA and DNA, basically untangle them, and it basically ends transcription. The RNA polymerase falls off, and now everything is separated. So that's row dependent termination. And then guess what? There's row independent termination, which means that we don't use row to do all of these things. And so there's no row involved. And so here, what we're going to do is the RNA polymerase is making RNA. There's a little mRNA coming off of it. And there's going to be a point around, near the termination site where there's a very GC-rich region. So here it is. And what happens is that it kind of loops around itself and it creates what is called a hairpin. So we call this a hairpin loop when the mRNA kind of folds in on itself, and it's folding in on itself because the G's and the C's are like attracted to each other here. Okay. Then what happens is NUS-A, this protein, comes and kind of binds to the GC-rich area. And it basically halts the transcription. Transcription can't happen when all of this stuff is going on. And so then what happens is the whole thing just falls off. So the RNA polymerase comes off, the mRNA molecule falls off, everything falls off. And that's it. That's the end of transcription. So there's two ways that transcription can end, one that uses row and one that doesn't use row. Okay. So now let's kind of just quickly talk about some antibiotics. Antibiotics, again, target bacteria and prevent them from growing or kills them. And one way to target bacteria is by targeting their transcription. So there's these two antibiotics called rifamycin and rifampicin. These both bind to the beta subunit on the RNA polymerase, and it basically blocks the transcription. Basically what happens is rifampin binds here, and this is the exit point for the mRNA as it's being built. So you know how the mRNA is being built and it kind of comes off, kind of comes out of the RNA polymerase this way. So it kind of binds here and blocks the mRNA from coming out. And so this blocks transcription, and obviously the bacteria die. Actinomycin D also does the same thing, but it does it in a different way. It actually binds to the nucleotide itself of the DNA. It binds to in between the G's and the C's. And then because it's stuck there when the RNA polymerase tries to come through, there's like a giant, it's like a roadblock basically. Okay. So these are the different types of RNA that the bacteria have. So of course, mRNA is the one that's used to make protein. Or encodes the information to make protein, but bacteria also have rRNA, which is called ribosomal RNA, which is used to make the ribosome itself. So ribosomes are made up of a mixture of protein and RNA. So the RNA part is called rRNA. There's tRNA, which you probably know is the one that transfers the amino acids to the ribosome when proteins are being made. And then there's a bunch of other smaller ones, sRNA, tmRNA, catalytic. We don't really need to know that, but just know that RNA is not just mRNA, right? We also have ribosomal and we also have tRNA, which are super important when we talk about protein synthesis. Okay. So moving on, let's just really quickly talk about the RNA polymerase in archaea and eukarya, a little bit different. So both archaea and eukarya usually have 12 or more subunits instead of 5, so they're a little bit more complex in that way. And then there are no sigma factors. So the initiation proteins that eukaryotes and archaea use, they don't use sigma factors. Instead, they have this protein called the TATA binding protein, or TBP. So that's one of them. This binds to the TATA region on the promoter of a gene, basically. So similar to the sigma factor. And then there's also TFB and TFIIB. Which is here and here again, does the same role as a sigma factor in the way that it kind of helps the RNA polymerase find the beginning of the gene. So yeah. Okay, moving on to 8.3. This is now we're just talking about the genetic code. So this is a little bit of review. You should be familiar with this diagram. As you know, a codon is 3 nucleotides, and each of the 3 nucleotides codes for a specific amino acid. So hopefully you know that already. And then really quickly about tRNA and anticodons. So tRNA is this molecule of RNA that carries an amino acid, right? So the amino acid is here. This is the RNA, tRNA. The amino acid is specific to this tRNA, and the tRNA brings the amino acid to the ribosome so that the ribosome can use that tRNA for protein synthesis. And the most important part of the tRNA is the anticodon. So at the bottom of the tRNA is this anticodon, which basically matches the codon specific for this amino acid. So if we were to go back to the genetic code, for example, phenylalanine, the RNA codon is UUU, okay? And so then the anticodon would be AAA because those are the A is what matches with U. So the anticodon would be AAA, whereas this would be UUU, and that's how the tRNA knows to bind here specifically, and it's going to be bringing the phenylalanine amino acid. Okay, so let's talk about how this is produced. So, we know that this is just a piece of RNA, which is easy to make, but how does a piece of RNA get bound with an amino acid? When you think about it, RNA is a nucleic acid, and then this is an amino acid. So those are two different types of molecules when you think about it in biology. So how do we get those two to bind together? And so we use these molecules called amino acid tRNA synthetases. Basically, what they do is they attach an amino acid to the tRNA. So how does this work, basically? So an amino acid is floating around, and it's going to attach itself to an AMP molecule. So here's ATP. The ATP is going to be broken so that the two phosphates are removed. And so now it's just adenosine with one phosphate, and it attaches itself to the amino acid, okay? By the way, these arrows are like used, if you remember in organic chemistry, like nucleophilic attacks, that's what they're showing, is the movement of bonds and stuff like that. But anyway, all you need to know is that ATP is broken and the AMP, which is the adenosine monophosphate, attaches itself to an amino acid. So here it is, the amino acid attached to AMP. So we call that amino acid AMP. And then this will attach itself to the tRNA. So this is the tRNA. And they're just showing that this is like most of the tRNA, but we're just looking at the very end of it, very tippy, the top of it. And of course, because it is RNA, we have a ribose sugar, right? The R in RNA stands for ribose. So here's the last ribose sugar in the tRNA. What happens is that OH group in that ribose will bind to the amino acid. It'll basically do a replacement reaction, and the ribose will bind to the amino acid and basically kick off this AMP. So this binds here, and then the AMP gets kicked off. So essentially now what we have at the very end is an amino acid, which is this guy, attached to the ribose, okay? And then this ribose is, of course, part of the entire tRNA molecule. And the binding is via an ester bond, okay? So that's really important to know, is that the bond that's created is an ester bond, okay? Cool. So now let's move on to talking about the actual ribosome. So as you can see, we're getting closer and closer to talking about protein synthesis. And so the ribosome in bacteria is very unique. It has this peptidal transferase activity. So that means that that's the enzyme portion of the ribosome that creates the actual peptide bonds. So that's super important. The bacterial ribosome has two subunits. We have the 30S subunit, which is the smaller unit. It's made up of 21 proteins and a 16S RNA molecule kind of mixed in there. And then the larger subunit has like 33 proteins and then two rRNA molecules, the 5S and the 23S. So there's a bunch of S's and there's a bunch of things being mixed in. Anyway, the small and the large subunit, when they are together, we call it the 70S subunit. Something to keep in mind also is that in eukaryotes, the ribosome is different. They have a 40S, we have a 40S subunit and a 60S subunit, which together make 80. So it's different from this. Okay, so now that we've talked about the ribosome, we've talked about the tRNA, we've created the mRNA, let's actually go into protein synthesis. So first part is initiation. So the first thing that happens is there is going to be an initiation factor number 3. So IF3 is going to come and shove itself into the E region of the 30S subunit. So something to also keep in mind is the 30S subunit has 3 little pockets. There's an E, P, and an A pocket. So the IF3 shoves itself into the E pocket of the 30S subunit. And what that does is it prevents the 50S subunit from binding, because the 50S is kind of on top. The way I like to think about these two, by the way, are the 30S subunit is like the bottom of the hamburger, like the bottom piece of the bread of the hamburger, and then the 50S is like the top of the hamburger. So you have like the top bun and the lower bun. And so the 30S is the lower bun. This IF3 binds to the E region and kind of prevents the top bun from attaching. Okay, that's good. So now that the 30S subunit is open, IF1 comes and shoves itself into the A pocket. Okay. And the mRNA comes and it binds to the ribosome. And the way it binds is that the ribosome binds to this region called the ribosome binding site, also known as Shine-Dalgarno. It's a specific site that the ribosome recognizes. And it's named after these people, Shine-Dalgarno. But anyway, so the 30S subunit binds to the ribosome binding site, and IF1 binds to the A site. So now we're here. It's looking like this. Okay. So as you can see, the only pocket that's left open is the P pocket. So IF2, okay, so this initiation factor 2 comes and attaches itself to the methionine tRNA. Remember that methionine is always the first amino acid for any protein because it's the start. It attaches to the start codon. So anyway, so this methionine tRNA comes and it binds to the P pocket. By the way, this is the only tRNA that can directly bind to the P pocket. Almost all tRNAs bind to the A pocket, not initially the P pocket. So this is the only one that can actually do that. But anyway, so now that the methionine tRNA is stuck here, IF2, it also comes with GTP. So GTP, similar to ATP, it's like energy, right? Okay. Anyway, so this this whole complex is now created. And then what ends up happening is IF2, 3, 1, 2, and 3, they just leave, okay? So everything just leaves. It uses GTP hydrolysis to do this, basically. So GTP, which is a molecule of energy, hydrolyzes, and this basically just pushes off IF2, IF1, IF3, they all fall off, which is good because we don't need them anymore because we have the first amino acid here. So we're ready to go. And so after IF3, IF1, and IF2 leave, then the 50S subunit comes. So the top bun of the hamburger comes and it sits on top. And now the ribosome is ready to go. So it's kind of like making a sandwich, right? You gotta have your bottom bun first, and then you got to, well, you know, like if you were to buy store-bought hamburger buns, they usually come like this, right? And so you take off the top bun, then you add the stuff, right? You add your, I don't know, lettuce and your meat and your tomatoes, and then you put the bun back on. So it's pretty much the same thing. And so now that we have the sandwich, we're ready to go. Okay. So then we're going to talk about elongation. So how does this protein get bigger and bigger, right? Because right now we only have one amino acid, which is methionine. So we obviously need more. And so there's more proteins involved, really annoying. So there's this protein called elongation factor that comes with another molecule of GTP. Remember that we need energy every time we do this. So the GTP is being brought by these proteins. So it's called EFTU GTP. So that's the elongation factor that comes with GTP, and it binds to the an incoming tRNA molecule. So another tRNA molecule that's coming in is going to bind to this elongation factor. Okay. The tRNA will shove itself into the A pocket or the A site. And what ends up happening is the GTP is hydrolyzed into GDP, and that, of course, releases energy. Okay. And then the elongation factor, now that it's done its job, it leaves. Okay. So now, right now, what we have in this ribosome is that first tRNA molecule and that second tRNA molecule with their amino acids kind of sticking out at the top. Okay. Then the ribosome itself will produce a peptide bond between the amino acids. Okay, so a peptide bond is produced, and that attaches the amino acids. And then what ends up happening is the connection between this, here, let me zoom in really quick. The connection between this amino acid and this tRNA is severed, so that now it's only attached to this second tRNA. So notice that this bond here is broken, and so now it's just this amino acid attached to this amino acid, which is attached to this tRNA. Okay, instead of them being attached like this, like a bridge. Okay, so that's that. And then another elongation factor comes by. So this one is elongation factor GTP. So again, coming with a molecule of GTP. And that GTP is cleaved so that energy is used. And this time the energy that's used is to create the shift. So let me go back to zooming in. So now that we're here, right, we want to add another amino acid. So this was the, oops, sorry, we're here, right? So we were here. This is the first tRNA, this is the second tRNA, and these are the two amino acids that have now been linked together. Now, if we want to add another amino acid, we have to expose the A pocket, because the A pocket is the only site where tRNAs can come in. And so what's going to happen is these two have to shift over one, right? So this tRNA needs to move over here, and this tRNA needs to shift over here so that another tRNA can come in from outside and bind to the A pocket. So basically what happens is this EFG GDP comes with its GTP. It comes, and the GTP is hydrolyzed, which releases energy, and that energy that's released basically pushes these two, and they both shift over. So as you can see, this tRNA has now moved to the E pocket, and this tRNA has now moved to the P pocket. Okay, cool. So that's that. Okay. And then at the very end, this entire EFG protein leaves, and so now notice that the A pocket is exposed, and we're ready to introduce a new tRNA molecule here. And also this tRNA, it doesn't have any job anymore. It doesn't have anything to do, so it's going to leave as well. Cool. And so that's basically how it resets. And so now we're reset, and now we have a tRNA here. This is going to be empty. This is also empty, so we're back to part one, and this will repeat over and over again until however many amino acids needs to be added. Okay. Last part is the termination. So at the very end, we need to reach the stop codon so that the ribosome says, okay, we're done. So first thing that happens, guess what? There's more proteins involved, right? So when we reach the stop codon, there is a release factor, and it's going to be either factor 1 or factor 2. It just really depends. But one of these factors will come and it will bind to the A site, okay? So the A site, the release factor binds to it. Okay. Cool. And what ends up happening here is that the protein itself, you know how there was like a growing protein, the whole protein leaves, okay, it gets cut off and it just floats away. Okay, so now all we really have to do, so the protein is done being made. All we really have to do now after this is reset the ribosome. So the rest of these steps have nothing to do with the protein. We are just resetting the ribosome. Okay. So RF1 or RF2 will come, and it will, again, help cleave the protein off. So now the protein is flying away. And so now we have to reset. Okay. And so what happens is another guy, RF3, so release factor 3 comes in, and it binds to the A site, which pushes off RF1 or 2, depending on who was there. Okay, remember it's RF1 or 2, so in this case, it was RF1, and so RF1 leaves, and RF3 is there now. Okay, not really sure why that had to happen, because guess what? RF3 gets kicked off in the very next step, so kind of ridiculous. Anyway, so this protein RRF, which is the ribosome recycling factor, along with EFG, remember EFG from before, the elongation factor? EFG with the GTP come in, and they both bind to the A site, and they kick off the RF3. So what the heck was RF3 doing that whole time? I have no idea. Anyway, but that's just the sequence. So first it's RF1 or 2, then it's RF3 who goes into the A site, and then RF3 practically does nothing, and then now EFG and the RRF come in and bind to the A site. So everyone is just coming into the A site and doing random things. Anyway, so now we're here in this sequence. And guess what? It's brought a GTP, and the whole point of GTP is to bring energy, right? And when this GTP is cleaved, the energy is released, and it causes the 50 and the 30S subunit to fall off, pushes them off of each other. And then IF3, do you remember IF3 from the very beginning? That's the initiation factor 3. It comes in, binds to the 30S subunit, and we're now reset. And so now this looks exactly like it did 3 slides ago. So let's go back. So now we're back here. Amazing. Okay, so that was protein synthesis. There is a video that I'm going to post on Canvas from the publisher that goes over all of these steps, and it's nice and animated, so definitely recommend watching that. Okay. So now let's move on to some antibiotics. So just like how we went over antibiotics that prevent RNA synthesis, we are now talking about antibiotics that prevent protein synthesis. So some of these are, so for example, streptomycin, it binds to the A site. Chloramphenicol binds to the P site. So they typically just bind to the ribosome and prevent protein synthesis by blocking the different sites. So tetracycline, again, binds to the A site. This one binds to the P site. So cool. Okay, bacteria are very efficient, by the way, when it comes to making protein. So when they make one mRNA, that mRNA is used many times to make many proteins, many copies of the same protein. So really efficient. So a polysome basically means that we have one mRNA molecule and there are multiple ribosomes using that same mRNA molecule at the same time. So here is an mRNA molecule. Look how there's 2 ribosomes. There could even be 3, 4, 5 ribosomes. They're all using the same mRNA, and the same protein is being made instead of just once. It's being made 4 or 5 times at a time. So it's just much more efficient. So we call those polysomes. Now, there's 2 variations of a polysome. You can have a polysome where this is just the mRNA. Notice that this is loose. The mRNA is already fully mature, and 2 ribosomes are using it. But another way that it can happen, and this is kind of wild, because there's no nucleus, when DNA is being used to make RNA, because there's no nucleus, the ribosomes have complete access, right? So in eukaryotes, because we have a nucleus, when RNA is being made, it's inside of the nucleus initially, and then the RNA has to leave the nucleus to find a ribosome. But in this case, because there's no nucleus, a ribosome can just come along while the RNA is still being produced, and the ribosome is already making the protein. The RNA hasn't been finished yet. Like the RNA polymerase is still finishing off this mRNA molecule, but the ribosomes are already using it. Again, very efficient. Bacteria can do this. And of course, it speeds up everything. And because bacteria don't have introns, it makes this possible, right? In eukaryotes, because we have introns, we have to do splicing after. But bacteria don't splice their mRNA. Once the mRNA is made, it's ready to go. And so as the mRNA is being made, the ribosome can already start using it, which is really crazy. Anyway, and so these 2 events happen at different locations. So this, if we're near the nucleoid region where the DNA is, then this situation can happen where the ribosomes are already using the mRNA before the mRNA has been fully completed. Whereas if you're away from the nucleoid region, for example, at the ends of the bacteria, then you'll typically see this, where you have ribosomes using the mRNA that's already been fully made. Pretty cool. Okay, after the protein has been made, there are some modifications that bacteria can do. It's not common, but they can do it. And again, these are called post-translational modifications. Basically, what they do is they attach different functional groups to the protein to, change it up. So glycosylation is a really common one, or methylation, adding a methyl group. Phosphorylation, obviously, we phosphorylate proteins all the time. So these are all modifications that can happen. Okay, proteins also need to fold properly. And so we're going to talk about how they fold properly and also how bacteria degrade those proteins in case the protein is screwed up. So there's 2 proteins called GRO-EL and GRO-ES. These are 2 chaperones. Chaperones are proteins that help other proteins fold. And so, yeah, those are these 2. So GRO-EL, and then this is GRO-ES. GRO-ES is basically the cap. We like to think of this like a trash can. So here's the bottom of the trash can, and here's the lid. And together with ATP, they can help a protein fold properly. There's another protein, other two proteins that are also chaperones, DNA K and DNA J. They also help with folding a protein. And then finally, if we needed to get rid of a protein, we have these CLP proteases. So I like to think of it as clip. Clip means to like clip or to cut, right? And these proteases basically cut and degrade the protein, especially if the protein is really old or if the protein is not needed anymore. Okay, so let's talk about how this all kind of comes together. So this is a ribosome and the, protein is being made and the protein needs to be folded. So the first thing that usually happens is DNA K and J come and they attach to the protein and they try to fold it properly. Now, if the protein folds properly, then fantastic, that's great. But if it still doesn't fold properly, what will happen is DNA K will bring it to the GRO-EL complex, and GRO-EL will take it inside and attempt to remodel it, attempt to fix the protein if it's not shaped properly. And if it is shaped properly, then fantastic. And of course, this uses ATP. Notice that DNA K and J don't really use ATP. It's only the GRO-EL that does. Okay. And then finally, if this still doesn't work, if the GRO-EL is not able to fold it properly, then the whole protein gets sent to a protease to be degraded. So that sucks. Okay, last thing we're going to talk about is what happens to proteins that are destined to be exported out of the cell. And so there are some proteins that are embedded into the cell membrane, some proteins that are sent to the periplasmic space if you're gram-negative. There are some proteins that are sent completely out of the cell. So how do these proteins get exported out? So let's first talk about membrane proteins. How do these proteins that are designated to be in the membrane, how do they get there? So there is some kind of signal recognition particle. It's a protein that binds to the protein. Anyway, so it's a signal recognition particle. It kind of just recognizes that, hey, this ribosome is producing a protein that is destined to be in the membrane. And what it does is it causes the ribosome to stop. The ribosome is like, okay, fine, I'm going to stop. And notice that it binds to kind of like the top of it, kind of like here. So this is the signal recognition protein binds to the ribosome that is actively making this protein that is meant to be in the membrane. So anyway, the whole ribosome pauses and the whole unit is brought to the cell membrane and it's going to go to this protein called FTSY. So FTSY is found in the cell membrane. And when the ribosome gets there, with the signal recognition particle, right? So these three are kind of all bonded together. So ribosome, the protein that is being made, the signal recognition protein, and the FTSY are all kind of linked together. So now the ribosome is given the allowance to continue making the protein. And so there's actually two options here. So we're either going to go up here or we're going to go down here, one or the other. So either the ribosome comes up here and the protein that's being made gets embedded into the cell membrane, and that's pretty much it. So very simple. But depending on the protein, maybe it needs a little bit more help. And so this protein called SEC-YEG will help embed the protein into the cell membrane. Again, it just really depends on the protein. But overall, the signal recognition protein is the one that kind of stops the ribosome and brings everyone over here so that the protein can be made directly into the cell membrane. Okay, cool. Another way that proteins can be sent to the cell membrane and even out is through the secretion pathway. So this is a very well-known pathway. So anyway, so protein is made, and while this protein is made, it will bind to this other protein called SEC-B. Okay, notice that this protein has not fully folded yet. It is just kind of wrapping around the SEC-B. Okay, so our new protein is the green one, and it wraps around the SEC-B protein. Okay, it's very important that this protein doesn't fully fold, because when a protein is fully folded, it's very bulky, and that makes it very hard to transport it. And so it's important that this protein finds a SEC-B and wraps itself around it so that it can be easily transported to the cell membrane. This whole complex gets taken to SEC-YEG as well as SEC-A. So SEC-A and SEC-YEG are hanging out here waiting, and this whole complex of SEC-B with this protein, the green protein, is going to come over and they're going to hang out. Okay, cool. And then what happens is the sec-A uses ATP to shove it through, basically. That's really what's happening here, is ATP is being used, and the protein that is supposed to be exported is being shoved through the sec-YEG channel. So that happens, and the whole thing is being moved. By the way, there is a signal sequence. So at the very beginning of this protein, there's a sequence on the protein that is called the signal sequence, and that is what tells all of this mechanism that this protein needs to be exported. So it's called a signal sequence, meaning it's signaling, hey, I need to be taken out of the cell. Okay. Anyway, at the very end, once the protein is released out of the cell, this other protein called LEP-B comes and removes the signal sequence, because the signal sequence doesn't have anything to do with the protein itself. It just has to do with making sure that this protein gets to the cell membrane and gets exported out. And so at the end, when the protein is exported out, the LEP-B removes the signal sequence, so now it's just a protein. Cool. And so if this was a gram-negative, this would be in the periplasm, and so maybe this is going to hang out in the periplasm. If this was in a gram-positive, then this just goes right out. Yeah. Okay. Next, a different way to export proteins to the periplasm. So in this case, the protein is already fully folded. So in this previous one, the protein was not fully folded, but here the protein is already pretty much folded. So a protein that is completely folded is inside. It has, again, this signal sequence. This signal sequence brings the protein to these two guys in the membrane called TAT-B and TAT-C. Okay, and then it's going to come and it's going to bind basically like this. Okay, now when this binds, it triggers a different protein, TAT-A, which is up here, to basically come over. Notice that there's like 1, 2, 3 TAT-A's, and they're all going to come over and they're going to assemble what is called a translocase. And the translocase is basically a pore that goes around the cell membrane, or kind of creates a pore into the cell membrane. Okay, so now that there's a pore, the protein will go through the pore and out. Okay, so now the protein has been secreted. And then that's it. And then it's released out into the wild. And of course, that signal sequence is cleaved off. So there's some kind of signal peptidase that comes and cuts off the signal sequence. So now it's just a protein by itself. Cool. So lots of ways to get proteins out of the cell. All right, last thing we're going to talk about is type 1 secretion system. So this is another way to, again, get stuff out of the cell. And this one is very specific to gram-negatives. And so it's this massive structure where we have an inner membrane ABC protein. So remember, an ABC protein is an ATP binding cassette. So down here is your ABC protein in the inner membrane. And of course, as you can see, it's using ATP to push something out. Okay. We also have this large outer membrane barrel. So this protein right here is a gigantic barrel that goes through the outer membrane. Okay. And then finally, we have a periplasmic protein, some little guy over here that's found in the periplasm that kind of holds both of them together. And so, let's say that there's hemolysin. This is just an example. This is a protein that needs to be exported out. Instead of having to export it out of the inner membrane and then through the outer membrane, this channel just lets it go straight through. So it doesn't even interact with the periplasmic space. It just gets funneled right through both membranes. And so ATP is used, it pushes it through, and then it goes out the barrel, and then it's out. Yeah, pretty cool. All right, so like I said, there are videos that I'm going to post, supplementary videos for all of this stuff, for a lot of the stuff in this chapter. So for the protein synthesis, there's a video, and then for some of this secretion stuff that we just talked about at the end, there are also videos about that. So definitely watch those. All right, thanks for watching, and I will see you in the next video.